Hi everyone, welcome to my presentation on local resting and action potentials. The first few slides, I am just going to be providing some base knowledge. An electrical potential is a difference in concentration of charged particles between one point and another. If there's no difference in charged particles between 2 points, then there is no electrical potential. Electrical potential is a form of energy that, under the right circumstances, can produce a current. An electrical current is a flow of charged particles from one point to another. Electrical signals are disruptions to the membrane's polarized state and the resting membrane potential. The electrical signals involve movements of sodium and potassium ions between the extracellular fluid, which is the ECF, and the intercellular fluid, which is the ICF. These electrical signals are triggered by stimulation such as chemical, light, temperature change, pressure, etc. The sodium and potassium gates are heavily involved in this exchange of ions, and the role of changes is mostly carried out by sodium ions, potassium ions, and cellular proteins. And you can see here that the sodium ions and potassium ions have a positive charge and the cellular proteins have a negative charge. The sodium/potassium pumps are constantly using ATP to maintain the resting membrane potential and the polarized state of the plasma membrane. The presence of sodium gates and the potassium gates in the plasma membrane provides a way for ions to move when a signal is desired. Gates in the membrane proteins that are specific to certain substances and can be in a closed state in which substances cannot diffuse through, or they can be in an open state in which substances can diffuse through. So in this next slide, I'm going to be talking about polarization and depolarization. Polarized is a state that describes the plasma membrane when it is at rest and the intercellular fluid is at negative 17 millivolts. Depolarization describes a shift in electrical potential across the plasma membrane to a value less negative than the resting membrane potential. Associated with excitation of a nerve or muscle, it describes the state of the plasma membrane at any time it experiences a shift in its voltage in a positive direction. When a dendrite is stimulated, its sodium gates will open and the sodium ions will diffuse into the intercellular fluid. This disrupts the negative 17 millivolts, causing it to become more positive. This shift into the positive direction of charge is called depolarization. So the first state that I'm going to talk about is the resting membrane potential. This is a state of being when a neuron is at rest and the intercellular fluid has a value of negative 70 millivolts. In this state, it's at rest and it's ready to send a signal down its length. A resting membrane potential is a stable voltage across the plasma membrane of an unstimulated nerve or muscle cell. The reason a cell has a resting membrane potential is that the electrolytes are equally distributed between the extracellular fluid and the intercellular fluid. A local potential is when the stimulation of a neuron causes local disturbances in the membrane potential. Typically, this response begins at a dendrite, spreads through the soma, travels down the axon. At the end of the axon, the process and end at the axon terminal. That is the order of the process. Depolarization occurs and the sodium gates open and sodium ions begin to diffuse into the intercellular fluid at the site of the stimulation, which causes its neighboring sodium gates to open as well. Local potential is produced by gate channels on the dendrites and the soma. It may be positive depolarizing or a negative hyperpolarizing voltage change. Local potential have the following characteristics. They are graded, meaning they vary in magnitude depending on the strength of the stimulus. The more stimulation that occurs, the more the sodium channels will open and the more the sodium ions will enter the ICF, depolarizing the membrane to a greater and greater degree. They are decremental, meaning that the current gets weaker the farther away from the site of stimulation it gets, and they are reversible, meaning that if stimulation stops, the membrane will return to its resting membrane potential. This is due to the sodium gates closing again without stimulation, and the pumps can return the sodium ions to the extracellular fluid. So lastly, I'm going to be talking about action potentials. These occur when a rapid change in which a plasma membrane briefly reverses electrical polarity. It has a self-propagating effect that produces a traveling wave of excitation in nerve and muscle cells. Action potentials occur only when there is a high enough density of voltage-gated channels. Action potential is a rapid up and down shift in voltage. It has effects that are great distance from its point of origin, and it always begins with depolarization. So to help understand the process of an action potential, we will consider the state of the gates, the movement of the cations, and the measured value of the membrane potential and the state of the membrane. So the first thing to consider, when a local potential from the soma reaches the axon hillock, it will begin to depolarize the hillock's resting membrane potential. It must be strong enough to cause enough sodium gates in the hillock to open and the membrane potential to reach the threshold for creating an action potential, negative 55 millivolts. Once the threshold is reached and the abundant gates in the membrane of the hillock open rapidly, and the sodium ions from the ECF diffuse into the ICF, this causes the membrane potential to undergo a rapid depolarization that we will see the value of the intercellular fluid go up from negative 55 millivolts to positive 35, or negative 55 millivolts to positive 35. At positive 35, which is the peak value of action potentials, the sodium gate closes and the potassium gates open. Now the potassium ions in the intracellular fluid diffuse out of the extracellular fluid. This causes the membrane to undergo a rapid repolarization, a shift of the voltage back to its negative values. When a negative value of 70 millivolts is reached, the potassium gates close. With both gates closed, the sodium/potassium pumps return the cations to their original locations, allowing the membrane to recover and to be ready to do it again. Some characteristics of action potentials. They follow an all or none law, which means if a stimulus depolarizes the neuron to reach threshold, the neuron fires at its maximum voltage of 35 millivolts. If threshold is not reached, the neuron does not fire at all. Above threshold, stronger stimulus don't produce stronger action potentials. Thus, action potentials are not graded like local potentials are. They are non-decremental, which means that they don't get weaker with distance. The last action potential at the far end of a nerve fiber is just as strong as the first one in the trigger zone, no matter how far away. For example, even in a pain fiber that extends in your toe from your brainstem, it's just going to be as strong. Action potentials are irreversible, meaning once they reach threshold, it goes to completion. It can't be stopped once it begins. Thank you guys for coming to my presentation on the action, resting, and local potentials. I think it's a complicated concept to grab, but I thank you all for listening in, and I look forward to watching your presentations. Thank you.